Welcome dear viewers to the video on dissolution release models, and in this video we are going to see the overview of drug release kinetic models. So dissolution models are the mathematical frameworks used for the understanding for how the drug is released from the formulation, and these models are very important to have understanding about the prediction for the drug release from the formulations, and particularly these help to understand about the relation between dissolution and bioavailability, therapeutic effectiveness, and thus help for the formulation optimization. So mainly the dissolution models are used to predict the drug release, formulation development. Then these are also required for the regulatory requirement fulfillment to demonstrate like bioequivalence, and then these are also used to have understanding about the IVIVC. So various models are there like zero order, first order, Higuchi model, Hixon Cal model, Crossme model, Weibull model, Packer Lansdale model, Hoffenberg model. So these models describe how a drug is released from the dosage form, whether the dosage form may be tablet, capsule or pellets into the solution, and in body how the drug is released into the gastrointestinal fluids. So that is the important phenomena behind using these dissolution models. Then this phenomena is predicted by using these mathematical models. These models help for the understanding of the drug performance in the body, for predicting the bioavailability and the formulation optimization. So first is zero order release model, and as you can see in the graph, this model gives the linear relationship between the time and the drug release. So drug release rate is constant over time, independent of the concentration, and it is ideal for maintaining the steady drug concentration in the blood. So always the zero order drug release is very difficult to have by the formulation design. There is a linear relationship between the time and the amount of drug release. So if you know about the osmotic type of formulations, you will understand that osmotic type of formulations, osmotic type of formulation design, or the osmotic release mechanism can give you the zero order release, and that's why the osmotics are popular. The present graph for zero order release shows a typical linear relationship between time and the cumulative amount of drug release. The drug is released at the constant rate and regardless of its concentration, which results in a straight line when it is plotted for time against the drug released. So every time interval you will get the same amount of drug release. Then coming to the first order release. First order release, the drug release rate is concentration dependent, and the drug release decreases exponentially with the time. This first order release is often applied to the water soluble drug formulations, and in the first order release model the drug release rate is concentration dependent. So it means that more drug is released initially and the rate is decreased over the time. So clearly it can be seen from the graph, which shows exponential decline in the amount of drug release over time. So initially drug release will be faster and then it will be de decreased. So this decrease is exponential decline in the drug release. Then Higuchi model. So Higuchi model describes the release of drug from the solid matrix based on diffusion. So mainly hydrophilic matrix tablets shows this type of or follow the Higuchi model. It describes the release of drugs from the solid matrix based on the diffusion. It follows Fick's law of diffusion and square root relationship between the drug release and the time. Higuchi model describes the drug release from the solid matrix, assuming that the release is diffusion driven. So mainly Higuchi model is based on the diffusion mechanism. The cumulative amount of drug release is proportional to the square root of time, resulting in a square root relationship in the graph. Then Hixon model. Hixon model describes or it accounts for the changes in surface area and particle size during dissolution. So mainly you can understand the erosion mechanism for the formulation if it follows the Hixon-Crowell model. This model is used for formulations or dosage form that erode over time, and this erosion is proportional to the surface area of the drug particles. So the particles may be drug particles or polymer particles or mixture of both, or these may be the pellets. The Hixon-Crowell model accounts for changes in the surface area and particle size during drug dissolution. It describes drug release is proportional to the cube root of the remaining drug amount. The relationship results in a linear graph showing decreasing drug release as the surface area reduces over time. So this is mainly the erosion mechanism. Then Crossme model, it is a semi-empirical model used for drug release from polymeric systems. It determines the type of release mechanism, whether it is Ficknian or diffusion. Then it is a versatile model for polymeric systems. The Crossme model is widely used mathematical model that describes drug release from polymeric systems, and it can account for various release mechanisms including Ficknian diffusion and non-Ficknian transport. Then Weibull model. So you might have heard about the Weibull model. Weibull model describes various release profiles exponential to sigmoidal, used for empirical fitting and flexible for modeling different dissolution behaviors. So as per my knowledge and as per my experience in the pharmaceutical field, Weibull model can be applied if your formulation like tablet formulation shows a strength dependent dissolution, or sometimes the tablet weight dependent dissolution, or the difference in the dissolution rate between the different strengths. So this is regarding the Weibull model use. The Weibull model is a flexible and widely used model to describe various release profiles in the dissolution, ranging from exponential to sigmoidal shapes. And it is mainly or especially useful for fitting empirical data that can capture different type of release kinetics based on its parameters. So this is like the for the Weibull model. Now, then coming to the Becker-Lansdale model, it describes drug release from spherical matrix. Modification of Higuchi model for spherical particles. So Higuchi model is modified and it is applied for spherical particles, and that becomes the Becker-Lansdale model. And it is suitable for spherical or semi-spherical matrix systems. So the Becker-Lansdale model is used to describe the drug release from spherical matrices, particularly systems where the drug is released via diffusion and matrix can shrink over time. So this is regarding the Becker-Lansdale model, mainly for the spherical particles. Then Hoffenberg model used for polymeric system that erode over time. So this is mainly the erosion based system model. It considers surface erosion during drug release from the polymers. The Hoffenberg model is used to describe the release of drugs from erodible polymeric systems. And also you can understand that whenever erodible polymeric systems are there, then there will be a surface erosion. So it accounts for surface erosion, and it is particularly applicable to the systems where the drug is dispersed in a polymer matrix that degrades over time. So this is regarding the Hoffenberg model. Now coming to the applications for model. Mainly these models are used for optimizing dosage forms which are designed as controlled release or extended release, or these are target release profiles. So sustained release formulations can be studied by using these models. Then a key part of dissolution testing in bioequivalence studies. You can apply these models to your reference formulation and test formulation, or you can apply for the formulation that went for the clinical studies or bioequivalence studies, and the modified formulation. So you can compare the test formulation with the reference formulation, and then you can understand that whether the release mechanism or release model is same or not. It helps to predict the in vivo performance based on in vitro test. So these are the applications. There are many applications also for the research field for these models. So this is the brief introduction and idea about these release models. I will make some more videos to have good understanding and detailed understanding about these models. So I hope you might have got a good idea about the dissolution modeling, and in upcoming videos we will learn in detail. Thank you for watching the video, and I request you to please do like, share and subscribe to Pharma Learning in Tech channel. Thank you.